We are hosting Her Majesty Professor Yaroslava Yingling from the Department of Material Science and Engineering, North Carolina State University. By way of introduction, Yara got her initial training at St. Petersburg Technical University. She was trained in computer science and robotics, computer science and engineering, to be exact. And then she moves to the state for her PhD at Penn State, where she got actually a degree in material science and engineering, and at the same time, a degree in high performance computing. So you can see the combination of material science and computer science take a very strong foundation to do what she's doing these days, as you will see. So then she did another postdoc at Penn State, working in chemistry, getting molecules, getting to know the molecules, and then another postdoc at NIH in nanobiology with a famous guy in RNA biology, Dr. Shapiro. And then after that, she joins the North Carolina State as a faculty, where she went through the ranks, and now she's actually a distinguished professor in material science and engineering. So Yara has a very interesting CV. She is, of course, very active, and as you will see, very energetic and engaging. But her group is applying multiscale modeling to soft and biological material in general. And she does everything. She does also structure prediction, enzyme engineering, and as you see, nucleic acids, and so on and so forth. So she has about 120 papers already, and since she has been in North Carolina State, she has brought more than $100 million money to if you count the grants that she has been involved in, including 5 major NSF and DOE centers. So I thought I had too many grants before I saw her CV, and I said, oh, too many grants. So she is also very engaged in material science and engineering. She is an editorial board, and including one of the chief editor positions in one of the material science major journals, as well as a founding chief editor and another one. It's the new one, correct. So yeah, so that I told you that she is working in multiscale modeling, and I mean, last time I talked to her at North Carolina State, they're doing a lot of different things. So, and she was kind enough to accept actually our invitation to come here, and it was a seminar with the title Molecular Modeling of Bioinspired and Biomimetic Materials, Predicting Stimuli-Responsive Behavior. Yeah.
Thank you so much. Thank you for coming. So I'll give you a little piece of our research, like one of my favorite babies in general, because I think everybody has a favorite baby. This is one. So I like the stimuli-responsive materials, the definition is pretty easy, right? Everybody knows what it is. The problem here, and a good problem for us as theoreticians, is they're supposed to have predictable properties. In reality, we are responsive material. Are we really predictable? Probably not, right? So I like to actually really focus on that. There's many different stimuli generally. We also have a favorite inspiration. I think every scientist should have an inspiration. My inspiration into this endeavor is the Venus flytrap. Why do I like it? Generally, has no central nervous system, has no brain. Somehow waits for the fly to touch 2 hairs, not 1, in order to close, right? So it has a memory over 35 seconds. So it has no central nervous system, no nothing, right? But there's a memory, and this memory is very responsive. It has temperature dependent, it has pH dependence. Generally, what's the 50 years of research into this topic, and I'm constantly interested and looking at it, produce that kind of picture where it's basically the trigger. So tiny change, basically it's change on an atomistic level, right? So if you just touch the little mechanical hair on the leaf, leads to growth and expansion of epidermis, and then swells up, we leave closed, generally due to ion transfer. So why would ion transfer happen here? Well, I think that's because of polyelectrolytes, but that's just my opinion, right? If I want to simplify something, right? So generally, that's something that we are very interested in. And why I like this topic, I'd like to make something like this synthetically at some point. I am an engineer, so I like to figure out what is the shortcut and what's a hack of the nature that I can put in order to make a material that's going to have responsiveness, going to have some interesting complex properties such as memory. And however, this is very difficult to characterize because it's enormous number of unknown variables, right? So the search parameters are very vast in general. In my group, we attack this problem on many different ways, right? So of course, responsive materials, we try to choose different responsive materials and try to find commonalities, right? So general. So we can take polyelectrolytes, what I call polyelectrolytes as nucleic acids, right? So general. LCST peptides or polymers that change their properties with temperature. Or sometimes we just add like magnetic particles, gold particles, all kind of different things to further control this type of thing. We take inspiration generally from biomimetic materials, right? So all kinds of different ones from cellulose synthase and transmembrane proteins predictions to like reflectin proteins to cellulose nanocrystals, anything that has an interesting type of property. Then we try to take this approach and put it into polymer composites and try to find the synthetic route to make something relatively simple. We call it design rules of materials, right? So we're trying to figure out that. And of course, that comes with properties on surfaces and interfaces. Again, we take some particles, we take some DNA, we start to mix it together and see, can we figure out, for example, this is a lot of work of us went into trying to figure out histone mimic nanoparticle that wraps nucleic acid around, right? So that's one of the work. And then we supplement ourselves with a lot of different things. We develop methods and software. I keep my hand in computer science all the time. So, we have this method called MD simulations. We have a software visualization. We're trying really to make in silico characterization tools for material science, mechanical properties, optical properties, all kinds of different things. And then the new method that we developed is all atom simulation with magnetic nanoparticles, for example. And we've been doing materials informatics for a long time. We just didn't call it that. We call it statistical approach. But really, that was machine learning, or if you want to call it materials informatics. Now we call it materials informatics, and we dab in it too. And I'll tell you what exactly we do because it's very specific. So as all of you, we work on 3 scales, 3 different types of things. I'm not an expert in DFT-QM. I'm a user. So it's expert comes here, right? So I develop something I know it's very well. Here I come to really take parameters from for force fields, like try to optimize these force fields and then try to noise these parameterizations based on the feedback from other things. What really separates us from a lot of other people is 2 things. One, we constantly validate and compare with experiments because I have a passion. My passion called data fusion. I'd like to integrate everything on one big data space so we can make predictions. So because of this, everything goes into heterogeneous data integration. So it's a lot of computer science. We use knowledge graph now, we use uncertainty quantification to really try to link properties that we observe from here on 3 scales and experiment. So we can use then materials informatics machine learning to develop better materials, for example, right? I guess I should switch now to more biophysics talk. So I gave a material speech, right? I'm gonna switch into what we do for biophysics. So I'll start with some very simple case. Polyelectrolytes, right? Polyelectrolytes, DNA, RNAs, generally polymer by polymer, carrying charged ionizable groups. The response to changes of pH, of salt concentration. What you see is a textbook scenario here, right? We genuinely say, okay, when we have a little bit of counter-ions, we're going to have extended or swollen material on a single level chain. We're going to have something like this. If we add a lot of salt, we're going to see condensation and precipitation, right? That's the general type of thing. We already have a lot of different types of materials that developed on that type of approach from what is called active materials, right, that can, polyelectrolyte gels that can undergo large scale changes in volume and shape as a function of salt and solvent quality and temperature, or we can make like capsules. This is PAA capsules. And we can see how the capsule change diameter as a function of pH, for example, right? So pretty cool things. What if we want to explain it and model it? Do we have a methods for this, right? So this is one of the questions that we ask ourselves. So as you all know, modeling polyelectrolyte is the biggest pain, right? So, or generally any charges, because they highly charge. So it's not that fun. The atomistic simulation is better, because generally we have chemical properties represented better, coarse grain. Okay, usually it's identical charge beads, and generally ignore all the chemical specificities, such as on top of the charges, it would be hydrogen bonding, directionality, all kind of different things. So the biggest pain for us, of course, doesn't matter what we use, this or that, we spend a lot of time calculating electrostatics. And so if you want to really model big type of materials, we would like to figure out how to go on a larger scale. That means we'll have to reduce something associated with this parameter. Don't worry about it, I'm not gonna write a new formula. Not that good. It's a different type of scale. So we decided to start with something simple, right? Let's see if we take DNA. In material science, we love it because of molecular recognition properties of base pairs. It's also well defined. It's highly charged polyelectrolyte and stiff polyelectrolyte. And in material science, we do all kind of crazy things with that, right? We can usually put things on another particle, so it's all the work, right? And we can make the one-dimensional, 2, 3, 4 all kind of multi-dimensional hierarchical structures with this molecular recognition properties of base pairs. We can also make capsules entirely out of DNA. What drives this self-assembly here? So molecular recognition properties will be driven, you can see here, by the single-stranded DNA, right? So the double-stranded is easy. What about single? So if I wanna look at the process of self-assembly, I need to figure out how to model single-stranded DNA, what's the properties of that? And that's what I like to do. So let's look at the properties of DNA. That's our beautiful picture. Everybody's seen this. People wear jewelry now, all kind of different things, right? This is everybody knows now. I think it's like biggest public outreach in general. So if we're looking at persistent lengths here, so persistent length is a basic mechanical property of a polymer chain. Generally, it is related to end-to-end distance, or radius of duration that you can measure experimentally. We also can determine it from experiments or a lot of different mathematical models, such as, again, we have different models here, persistent lengths. This is your intrinsic persistent lengths within the absence of charges, for example, for neutral molecule. And from here, we can figure out radius of duration. So that is one like chain model, where persistent length relates to the radius of duration. Alternatively, from simulations, the easiest way for us to calculate it is like this. All right, so that's much easier. Just go through backbone, calculate the angle, voila, you have a persistent length. So when it starts to turn on itself, you know this. So the persistent length, so it's basically the length of a stiff part of a molecule. For double-stranded DNA, it's very, very well known. Generally, it's 500 angstrom. It's been developed for a long time. So that's our double helix. However, for the single-stranded DNA, there's all kinds of different cartoons that people use. It's a known structure. Persistent length ranges anywhere between 7 and 20 angstroms in general. So it's a large difference here. If we want to look at the self-assembly, we will have to capture the stiffness of this molecule. Otherwise, we can't really form the self-assembly. And 7 to 20 angstroms difference in persistent lengths, right? It's like between a piece of paper and a table. I'm saying this is the same, right? So the equivalent is really hard. What can we measure it with? So for double-stranded DNA, you know, it's NMR and X-ray diffraction. But I don't talk about double-stranded DNA much. I talk about single-stranded DNA, flexible polymers. So there's different types of experimental tools that we have available to us, right? We can do FRET and measure end-to-end distance, right? That's the traditional formula. We can use SACs, measure radius of gyration. That's going to be our SACs profile. Gives you radius of gyration, gives you your shape, give you even the behavior on the interface, right? SACs profiles are pretty cool. And then we can do a course extension. So basically, you take the tip, you pull the DNA, and what you measure really is the base stacking, right? So there's 3 different ways. You might mention it because I'm going to compare to these techniques. What so far we know about single-strand DNA. So I started gathering this research in 2012. I took someone's review paper and then adding it into that paper. So the last paper shows the persistent length is right here, approximately for this salt concentration, for DNA between 2 origami pieces. Why is such a difference? Because let's take this 0.1 mole. We like using it in simulations. Usually this is our range here. So basically I'm saying that experimentally I can choose anything between 8 angstroms to 35. So my stiffness could be anything. It's just the playground for me. So I can choose, I'm going to parameterize based on this or this. So which one should I use? So I'm deeper and we looked at what is measured, what the sequence here. And so, for example, you can see on colors, based on colors, there's different measurements. So this one is SACs. So SACs are measured to RG, and using one-line chain model, you get persistent length. This is FRET and SACs. Here's end-to-end distance, right? So this is boom. Okay, here, this one is FRET, 10 to 70 mer, all the time in. This is force extension. This is mixed bases, very long force extension, and this is brand new, staying up a bit right here. This is measuring the distance between DNA origamis, right? So that gives us that persistent length varies on experimental technique, on a sequence and length, and notice formula, right? So for example, this is FRET, this is FRET, end-to-end distance, slightly different formulas, slightly different extension, slightly different error, right? So it's like everybody does what they want. And we see that the value of persistent length of single-stranded DNA is dependent on experimental technique, salt concentration, solvent quality, length sequence, and what else, right? So it's basically a really hard and interesting problem. So what we did, we decided, we're going to take this single-strand DNA, throw it into ND simulations, and voila, we can calculate persistent length directly from backbone, right? Question is, which force field do I take? What water model should I take? What salt concentration of parameters for salt should I use? So one of my grad students spent a couple of years doing this study, maybe even more, because we really scanned as many force field combinations as we can. So here you see a good case scenario. So what do you see here? So this is the range of end-to-end distance experimentally measured. This is the range of radius of gyration experimentally measured. So if we agree with experiment, we should say here, somewhere here in the middle. What we see here, different force fields, FF99, we went with amber, just in general. This is a new one, this is 0, this is BCS1, and this is OL15, right? So this is implicit solvent. Now, this is explicit solvent, the same force field, but now it's explicit solvent. We use 2 different ones. We use TIP3P and OPC. I think this is actually was 4 TIP3P because it's produced slightly better results. Okay, we can see generally speaking, all of the force fields behave pretty okay. They're not outrageously far away, right? So generally, I would say it's a success. However, we see that some force fields perform better. We wide agreement with experiment. So because of this, we say that we chose FF99. It's an old force field. It's a very old force field, and it performs better. Why? Why is new force fields don't perform better? Because we optimize it towards double helix. So FF99 doesn't matter how, not good for single, so double-stranded DNA. It's actually performs better for single-stranded DNA, but much worse if we do double-stranded DNA, right? So because all of the other ones is, it's improvements from the dihedral angles and biostability interactions to improve the stability of double helix. Okay, so this is the force field we took. And we took, for example, the polythymine We know theoretically that persistent length is a combination of intrinsic property, intrinsic persistent length, neutral. So how do you measure experimentally? You put a lot of salt in it, right? And generally you assume that, okay, you screen all the charges. So it should be smaller than your length of the charge interactions. And then we did, this is our old work, we actually changed the partial charges and then left the whole same architecture, but the partial charge is now 0, right? So we just scale it so it's not charged. We didn't want to cut anything, so this one you cannot make synthetically experimentally, right? And next one, we decided, let's look at the backbone. So from here, it's uncharged, so we should get very precise value of intrinsic persistent length. Now we cut off the bases, because bases are pi stuck, we don't need it. This one we left -1 charge, and we just parameterized with hydrogen here and just kind of left it exactly as it was with minus charge total. And then we also knock out the charge. So now we have two things that allows us to do simulations. So this one allows us to decipher contributions of persistent lengths from electrostatic interactions. And this one allows us to compare to a simple polyelectrolyte. It's very simple, still directional, right? So still directionality of the backbone, which is prominent for all biophysics versus simple polyelectrolytes. It still has the same charge. We also can determine intrinsic and electrostatic, but that should follow better generally all the simple polymer physics. So what happens? What happens? So what we can see here is persistent lengths. So this is the one that we measured from experiments as a function of lengths of the bases from 10 to 16. We see that initially we have about 10 angstroms, and we're suddenly raising up to pretty high lengths of thinking. And then it goes down and kind of levels off. If we're looking at L0, so this is L0, it's a contribution of persistent lengths if DNA is not charged, right? So this is our contribution, total contribution from charges. And then we can look at this by fitting different type of theories here. So we have L0. So now let's take Darwinian or Darwinian theory, which is new ISF theory, and just plug in the formulas back in. And we see that none of the theories, yeah, they're higher, persistent length is higher, but we don't reach this kind of big gaps. So in simulation, we have much higher persistent lengths generally. If we do it to the backbone only, so this is our intrinsic persistent lengths, this is our measured, this is our formulas that fits just perfectly well. If they would develop for this, we can see, yep, it works well. If you don't have basis, it doesn't work. The theories does not let us have a prediction. So what really happens there? Right, we went and looked through all of the little motifs that formed at the ends of the DNAs and deciphered them, what's going on there. And we can see again, if you're looking at number of total hydrogen bonds versus number of bases, is polythymine is orange and polyanti, it's neutral one, right? So that still forms the base pairs. We can see if we have charge on the backbone, we form more hydrogen bonds. We also form more pi-pi stacks. We also form more TT interactions. We also have generally longer average consecutive pi-pi stacks, one on top of another, right? So it gives us more stiffness to that. So we can see that generally we see the trends of our persistent lengths agrees with the pi-pi stacks and consecutive pi-pi stacks. So that's what gives us this hump that cannot be explained by experiments, but by theories. So we decided to look into what would be the difference in our DNA if we're going to look at it from the experimental point of view. So here are two ways to calculate radius of duration, right? One is directly from simulations. So it's a simple radius of duration from 1D, it's a black line. And another one, we're going to calculate persistent lengths and then put persistent lengths value into worm-like chain, privilege joint chain. Sorry, I've been saying it for much. It's a privilege joint chain model. And we put it here. So, and then this is RG. So we have RG here, right? So here we see that this is our true, and this is your RG after you calculate it through the persistent length. So this is disagreement, right? So we again see that we can't compare it to apples, orange to oranges. It's not really a good comparison here. If it's for the backbone, it's exactly the same, right? So backbone through RG calculated through persistent length. So our method is correct, because initially when I saw this, I thought we messed up on the formula or something, because how come, right? So no, it's for simple polyelectrolyte, which this was developed for simple polyelectrolyte, it's actually worked well. If we have complex polyelectrolyte with this pi stacking, consecutive pi stacks, hydrogen bond formation, it doesn't give us a good variation. So what we did, we developed a new formula. We took this, we scaled it, and shortened it so it fits right much better here together. So we basically descaled this formula and brought it back to true RG. And this is our formula that correlates radius of duration for Sachs, for example, with the persistent length. We did the same thing for end-to-end distance, right?
Could the difference be that the definition of monomer is not the actual
The definition of monomer here? We tried all kind of different things. We tried to do monomer. We even tried to do little loops, right? So looking at like a nodulus when they form this couple of base pairs and try to do this, so there's different ways how we try to assume this angle is between the monomers, rotation between the monomers versus like dimers, trimers, and so forth.
I mean, in this formula.
For this one?
No, the second from the top, this one, yes, it's tau, right?
Yeah, it's persistent length. And the persistent length is calculated from the rotation of the backbone. So we're going through the backbone, not the side chains. So generally that does not take into account the side chains at all, because it's only looking at the rotation of the angles in the backbone. And that's it. So, and this is just plugged in into Borland chain. Maybe I went too fast. All right, so what we got is new formula that can scale it a little bit higher. We still don't know the structure of the next thing. We decided to look at the Sachs because RG is related directly to small angle X-ray scattering, right? Sachs. And this is experimental Sachs for polythymine. And all of the fits that you see here is from our different combination of the force fields. So this is FF99 that I was just showing you, and then other force fields here. You can see that our simulations, this is snapshots over the course of 100 milliseconds. So we ignored the rest of it as it was folding, right? So we don't only look at the conversion. We can see, okay, sometimes we fit some interfacial area, but we miss the side. Some of them, okay, the interfacial area is incorrect, however, the size is good, right? So you can see that all of the place, we have not found one that really tells us we have a really good agreement with experiment. None of these force fields, even though they approximately work in terms of radius of duration, overall size, but that does not capture, again, here, it captures size, shape, and also captures the interfacial behavior. That's the power of Sachs, right? So we only capture RG. If we're looking at the snapshots, so this is, you can see it starts rolling from the kind of like buttons and then stays in this type of structure. Here, we very much form double helix. It's just folds on itself and form the helix because it was developed for helix. This one doesn't fold at all. Right, so we have all kind of different structures. None of them look any reasonable. But if you're looking at RG, yeah, we're close. Right? So what we did next? Machine learning, because finally we give up, right? We've gathered so much data, and now we can actually start manipulating our system and looking which snapshots would be the best one. Where's our ensemble average? Because when we're looking at a single simulation, we can't guarantee we sampled everything, right? So what we did, we dumped all the simulation snapshots there, treated them as individual population genes, right? And then we created chromosomes, took about 5,000 structures from the simulations, and then calculated from these chromosomes the Sachs fit, calculated the fit, the lower the fit, the better the value. Then did a couple of things. This is genetic algorithms, so we did elitism. So if the fit was very good, it goes to the next level of integration, or we also add some breeding and some crossover mutations, right? So we added some mutations to the genetic algorithm. Genetic algorithm is very popular in material science. What output did we get from that? One point, a couple of things. So this is one of the examples from our paper, but for example, that's the same thing. End-to-end distance, radius of duration. This was our simulations FF99. Now, what you see here in red is the chromosomes at the end that was chosen to have the best fit here. So you can see that they're all over the place. They're all over the timeline too. They can be chosen from anything. We started from 0. We actually calculate the whole thing, right? We modeled it. And the fit now, experimental versus computational, is just amazing, right? So GA chose the structures that represent the SAX profile based on simulations, the best, right? So structure still out, just the fit to experimental measurement. Look how cute they look, isn't it? Just asking for cover here. So that's kind of interesting because we didn't expect it. It's the best fit, of course. Is it correct? No, it's a prediction. It's still simulations. It still has limitation from the force field, but it gives us an idea of how it looks like. I was actually surprised. I still thought this kind of ants that roll in each other would be prominent. They're not prominent in this whole ensemble. They're one of the features, but not the whole ensemble. Right, so that's what I'm saying. This creates pretty ugly cover with single-strand DNA. I bet you can make better images than this. And here we have an information. Now we know the structure of single-stranded DNA. Approximately, we know this behavior, and we can approximate behavior of this as a function of salt with our new formula by converging Rg from SAX with our new formula. We have approximate behavior of persistent length is roughly proportional to salt concentration to the power of -0.2, right? So it's only SAX that we convert from, keep this in mind. So we can say we resolve the structure, even though it's a strong word. I always get necked in the papers. We predicted the structure. Even though we resolve the SAX curve with MD simulations, basically. It's not a new, by the way. There's a lot of things in material science when people try to use SAX. And so it's not a new idea. Genetic algorithm is a little bit newer side, but generally it's just implemented. We found the best combination of the force field that fits this SAX profile the best. And we found a general formula that does better job of connecting Rg to persistent length. Better formula, not perfect, because this is a number that we cannot explain. Right? It's a constant that's maybe sequence dependent, maybe length dependent.
Yeah.
We ran out of funding. That was done.
That was my question, actually. How do you deal with the sequence dependence of the persistent length, for example?
I don't deal with
You
this.
don't deal with
Once-
polytiming all
Polytiming.
Okay.
Almost all of the experiments are polytiming
Nice.
because it's easier to synthesize.
Yeah.
We did polyadenines too. It's still okay. There's a couple of papers on polyadenines, but cysteine and no. There's-
Cyclizine nice.
So if you don't
Yes.
have it, and it's generally becomes
Yeah.
much harder, right?
Mhm.
Okay, so now we want to again simulate the materials. We don't want to calculate electrostatics. We know general behavior and the persistent length of the DNA in solution, right? So we know what it does as a function of salt concentration. We now want to move into something like this, where we can see the change in materials properties as a function of salt concentration and pH. And look at the morphology. So we moved into different techniques. It's called the speedy particle dynamics. No one likes it except me. It's like a small community that likes it. Why people don't like it? It's like American high school, right? It's still Newtonian's behavior, but instead of a regular potential with equilibrium, everything in your system hates each other. And whoever you hate less, you kind of segregate, right? That's what I'm saying. My kids went through American high school. This is it. This is DPT technique for that, right? No, it's a joke, of course, but that's kind of gives you the idea. It's very simple. It's very quick. You don't have to calculate equilibrium. There's nothing in there that is required. What's the difference here? Well, we can scale this AIJ higher, lower, as a pair interaction. You hate one molecule less, another one more. Very, very simple. What is it for? Usually the techniques formulated by Warren, it's for coarse grain simulations of phase separation, basically, right? It's very quick, it's fast, it's generally parameterized up to density, volume fraction of 0.3. That's it. However, they all charge them. So how do we simulate polyelectrolytes with that? But we still don't want to do electrostatics because we want to make sure that it's not, we can figure out this somehow and feed it into a simpler system. What we did, we actually made an assumption that charged monomers are just self-repulsive, right? So they will be repulsive by themselves to each other. Then we can basically use the concept of second virial coefficient, which is inversely proportional to solvent ionic strengths, and parameterize the DPD exactly the same, right? So we have intrinsic contribution, electrostatic contribution, and electrostatic contribution is inversely proportional to solvent ionic strengths. So this way we have no explicit charges. We just added something that Ruth and Warren didn't add. That's all super simple concept. Genuinely, APP, this one between two different beads, will be inversely proportional to solvent ionic strengths because there's no explicit salt anymore. There is explicit water, but not explicit salt. And then high salt concentration will lead to basically neutral behavior of polymers, right? So it's like everything is screened completely. The advantage is large scale behavior. We parameterize this. So this is DPD. This is also, remember, we had LP proportional to solvent ionic strengths. This is Dobrynen's work on atomistic simulations. RG is proportional to simple polyelectrolyte to the same type of behavior. We see that we agree in general with atomistic simulation, not ours. It doesn't matter for even very long chain. So at low salt concentration, we have extended form of the chain. As the concentration increases, it's collapse. What we cannot do, there's a lot of limitation, and most of them associated with mean field theory. So it means that we ignore finite size of ions, could be a problem. There's no correlation between ions, no correlation fluctuation of counter-ion clouds. We can account for multivariate ions or very low salt concentration, and there is no attraction between two DNA, so we're not allowing the hybridization to occur technically. What we did with this, we did two couple of things. We started looking at DNA micelles. Why DNA? We had experimental collaborators ready to synthesize anything we're going to make. So we went with DNA, right? So we had hydrophobic block. So that was polymer tail, any type of synthetic tail. And this is DNA charge and amphiphilic, right? Technically, this can be used for drug delivery. Another one, then we added another construct. We added another tail on another side. So now we have a tri-block. Again, DNA is blue here, and then we have very different properties of the tails and lengths of the DNA. And it's this DPD simulation is extremely quick. That's actually not simplified, it's just everything collapses immediately right into the double structure. What do we got? We've got morphological phase diagram, again, back to material science. So in my morphological, everybody in material science would love phase diagrams. This is what we make us happy. So you can see this is the length of DNA, and this is solvent ionic strengths. So here we have this almost everything screen, so it's a high salt concentration. And we see as we increase the length of DNA, we go from vesicles to like pancakes to worm-like cylinders and then spherical polymers. And now the beauty of our work that we can predict what's going to happen to this material if I form it at the high salt concentration and then drop it to low salt concentration. I can see what happens to the cylinder that goes into spherical micelle, or this worm-like chain, the worm-like cylinder to also a couple of things. So we can scan all of this. We can also then quantify them based on anisotropy. Right? So this is our vesicle lamellar. It's also our region where we can call it precipitated. We only formed one structure in simulation. So technically it's an aggregate, it's just something, but it still forms structure, so we call it visible, right? So it's highly structured. Then above here, we start forming many of it. So it's not just one, it's one representative snapshot, but there's many of them in the box. It's 4,000 chains, so it forms pretty large aggregates, many of them. No one believed us when we did this because we did such a hard approximation. DPD is hard approximation already, and on top of this now mean field theory. So we asked our collaborator, Stefan Zauch and Torsten Polly, to verify this construct right here experimentally. It's not easy for experiments to do all of this. So what they did, they took the DNA poly-T to poly-T50, the same as us, and attached a synthetic tail. This is a structured synthetic tail. It also has fluorescein in it, so you can see it. And they looked at the FM, and you can see as ionic salt concentration increasing, we see that they are forming micelles, so they are forming something, but it's hard to actually gauge what exactly something formed. They success, no surprise. So the next thing we did, we compared our prediction for radius of duration. So this is blue with static and dynamic scattering. This is black. Yeah, so what was interesting here, first of all, is pretty good prediction. What we see here, this is the salt concentration. That's the size. We can see initially, when we have low salt concentration, we have a high repulsion, so chains are extended, corona is big. Then as we're starting to increase salt concentration, we're going to be naturally seeing smaller and smaller because it's kind of condenses on itself. And technically, if there is no morphology change, it should continue, right? If there's no morphology change. What we captured here for the first time is a change in morphology, right? Then, as Stefan said, that if you would see that without simulations, you would think it's just something strange because we didn't know that that's going to be signifying this type of transition, the change in morphology from spherical to a cylindrical. So anisotropic changed, and we see it here with the difference in the salt concentration. So our model is showing that we can explain experimental observation and quantify morphology and property change. As everybody else, again, once we did most of the work, we were very lazy. We just want to tell people the formula. This is your design. This is what we call materials design rule, because it tells you how your radius of duration will change as a function of salt and lengths, salt concentration and lengths of polyelectrolyte. Right, so radius or corona. So this is what we got from spherical micelles, for splitting. And then we compared it to different observations from different papers. So you notice that most of them are not DNA, but it really doesn't matter. We are within correct values. Height of the corona, you have minus 0.11. Here's minus 0.13. This is different one, minus 0.2. Okay, so we're right in between, so perfect. If we're looking at hydrodynamic radius scaling, again, we are close, right? So this Borisov and Julian theory, okay, we are parameterized based on their theory, so no surprise we match them. So if we don't match them, it would be pretty bad overall. From that point on, we created more simulations. We added other type of things. We increased the lengths, synthetic lengths. We made it stiffer. We played with the lengths, not just lengths of polyelectrolyte. This simulation, the whole morphology diagram can be created in 1 month. It's super, super quick. So we just did a lot of different other things. And we found more general formula now with the dependence on the synthetic tail, right? Stiffness of the tail and length of the tail. So now we have much more general design rule for materials prediction. That's ended Nans Lee and Thomas Liebman thesis. That's thing that you can put on a t-shirt after that, walk around this time. So we have excellent agreement. So from here, we went into something more complicated. Now it's gels, right? So you can make DNA gels, but it's now we have two different things here. Right? Since we have two hydrophobic blocks, we can form bridge chains, which relates directly to like cross-link and density, so mechanical properties of the gels, and loop chains. So it goes back into the same micelle. Basically terminates on the cell, right? very interesting behavior. We wanted to know morphology changes as a function of salt concentration. We also wanted to make sure that we understand mechanical properties. So that was two tasks that we wanted to do. So started with volume fraction of 0.1 and found an interesting type of behavior, completely different. Notice this is gel here, and now here we have a lot of different type of properties with different morphologies. My student created this. She was probably hungry, so she calls it raspberry particles. You see little type of things eating on a blue, vertical, but decorated with DNA. Then hamburgers, and you can see we was thinking of selling it to McDonald's as a franchise. Look at this double hamburger and triple hamburger, and even like something really complicated, like a roll, right? And then the next one is spaghetti and meatballs, right? So again, the roll of something and to the. So that's our phase diagram. So basically, again, below these lengths, we're forming one aggregate. So again, precipitate maybe structure. Hard to tell. With gels, there is another parameter, volume fraction. So that will change as a function of volume fraction. We actually specifically focused on this area because that only forms one particle and repeated it at different volume fractions. And we found this, I don't know what it is. It's some kind of mostly artifact, but it's really cool. And it forms like the core, hydrophobic core, forms like the spiral type of structures, right? We see it here. We kind of see it here with a triple hamburger, and we see it here. Generally, pretty boring things. Sometimes we form some organization, and most of the time we form the gel. That's it. So again, ethical Coley did a couple of checks for us. So she looked at polyelectrolyte named 30, and she used DNA for this transcript, so 2 alkyl chains and DNA in the middle. And then she changed the volume fraction and observed the same type of behavior at a higher volume fraction. This is it's hard to see this, but our sizes agree quite well. So you can see this is our hamburger. There's a couple of hamburgers here. This is our like worms here, these type of things. And here are the gels, right? You can see gels, and again, this type of interesting structure. So generally we have an agreement, so we can move on with our analysis. And what we did first, we wanted to see what happens with mechanical properties. So for example, calculated fraction of bridges. So it's cross. So basically, the more bridges you have, the better mechanical properties. And another one, we looked at domain size, right? So generally number of pores, because if we're using it for drug delivery, that's where you're going to be drugs. So if you separate into smaller pores, the diffusion is faster, the drugs dissolves out. And as a salt concentration. So what would be this as an equivalent? Oh, oops. That means once you put the equivalent, this thing will happen. I'll just play this. So you can see that generally, again, we have the gel that, so we start here. So we have the condensed particle with 2 pores. Initially, as we're starting to decrease salt concentration, we're going to see the change in number of bridges, and then it abruptly rearranges and becomes spaghetti and meatballs, right? So this is your recipe for creating spaghetti and meatballs out of hamburger, I guess. And if you're looking here, the number of pores, however, grew pretty steady as a function of ionic strengths. The mechanical properties, however, were discontinuous. And they really change from morphology. So we can capture experimentally the change in morphology with the mechanical properties, but not necessarily with number of pores. After that, we again went into doing all kind of different type of scaling, trying to find the correlation between parameters for number of pores. And we looked at number of pores versus different properties here for the prostatic interaction, with solid dynamic strength interaction and lengths, and was able to come up with a general formula overall, where a number of pores is directly proportional to volume fraction of the polymer, inversely proportional with the length of the DNA, inversely proportional to salt concentration. So we can do that. So that's easy, right? It was like kind of changing quite normal. And then we were very bold to try to do mechanical properties. We really noticed doesn't look good. It's not correlated. We saw though neutral polymers have two things, right? They have entanglement generally, and if we focus on the entanglement, we're going to see, that's going to be proportional to fraction of bridges and volume fraction to the power of 1. This is neutral polymers. It is textbook thing. I didn't come up with it. We wanted to make it for polyelectrolyte. We decided that elastic modulus is only going to be dependent on the number of bridges. And we were able to come up with a general formula with this interesting exponent, which depends on still polyelectrolyte length and salt concentration. It converges past 0.3 volume fraction into something, but not at the low volume fractions. You can see this is this exponent. Voila, we try our best. We published and said, someone else doing this, I'm tired. We spent so much time on it and we didn't get to that. But why is it we didn't get to this? Because mechanical properties changes a bit different. It's not easy to predict it with that. So after this, just one slide about this, change the topic to something even more exciting. We added in our system ability to form bridges. Not like permanent covalent interactions. So basically once it's an addition of kinetic multicarl equivalent into the dissipative particle dynamics. So once they meet the criteria for distance, type, and time, we can form covalent interactions, and then we can start looking at salt concentration versus hydrophobicity and see that at some hydrophobicity start forming these cool fibers. You can see like little interesting fibers formation. Our collaborator Kila Fonin was able to show this based on RNA fibers. So it's a bit different. I understand it's a long stretch. His interactions was, so this is like dumbbells, RNA dumbbells, and they were interacting, forming kissing loop type of interactions, forming the interesting type of fibers that weakly agree with them. But kind of an interesting idea. We also try to do the hybridization natural way like this. We were able to succeed, so that does not form permanent bonding. We changed the representation in 2 beads per monomer, and it was able to show hybridization of 2 strands together. That's all. But as soon as we put more, it created terrible mess.
So what do you do? Go from polythymine to this sort of hybridization of 2 strands?
So this one, we changed the, so now we have 2 beads per monomer here. So 1 bead is backbone, it's charged, another one is a base and can make a connection, preferable connection with another base. So this would be polythymine and polyadenine.
Okay.
Yeah.
I
So
see.
they, it's
Even though it's all based on polythymine, the development was done based on polythymine, but it's, okay. I see.
now we try to mess around with this, but
I
we
see.
still have persistent like mechanical properties based on
Right.
polythymine.
I
So we're just looking at molecular recognition properties of base pair, allowing them, so if they found the base pair, they can click and form a bond. This was very difficult. We was able to do just this, and this happens very quickly. It's kind of cool to watch it. But if you want to see material, it doesn't happen. This happens also very quick, but this is maybe an artifact because once as soon as the criteria is met, so for example, if I have 2 strands, they come here, criteria is met, the next strand is going to be like this. Right? So it's not like a natural that type of sliding motion. It's kind of a rough approximation. Nevertheless, it shows something possible from that community. I'm almost done, right? So modeling for electrolytes, we did all of this specifically for DNA because we were interested in single-stranded DNA structure and materials absorption. And then we realized that atomistic simulation is too short yet, so we went into really ultra-coarse grain model. Basically, DPD is worse than coarse grain molecular dynamic simulations, right? It has no equilibrium, but this allows us to get a really nice agreement with experiments in ethical cooling. We even try to put these micelles into the cell, for example. In the interest of time, I was going to tell you about responsive systems temperature, but I see I ran my mouse too long, so I'm going to skip a lot. You can ask me questions if you want. But basically, LCST interesting. Why? Because they do something that I was very surprised the first time I started working with them, like many years ago. Because I assumed that if you heat up everything, it's going to denature, right? This one, if you heat, they actually condense, right? So this is, it's the opposite. So you, as you're heating it, goes from transparent to opaque, right? So this is really cool. And this is elastin-like peptides. They have a very long sequence and structure and everything else. They form beta tones, that's it, basically, except the beta tones. There's not really secondary structure. And they use the material science. Two things to take away from this. So experimental observations show the LCST. They show a very specific drastic change in absorbance as a function of temperature, as a function of sequence. Super interesting things generally, right? Simulations of a single chain always showed radius of duration versus temperature, that type of behavior. But with polymers, yes, there's a different type of mechanisms there. We studied that. With the ELPs, that's just non-converged simulations. For example, different force fields, different water models. Which one does behave like this? Where is this? Nowhere to be found, right? If you simulate it for 100 nanoseconds, because this one was 9 nanoseconds, and this is 100.
9 nanoseconds.
Yes.
Oh, wow. I see.
I didn't put a reference here for you. You don't need to know that. But that was for a very long time. All the experimentalists was telling me, do that, show me that. Because this is, everybody knows that. Where is it? So this is it.
Okay, not
Here. We tried everything, right? So it's not a simple, okay, we can say, okay, here a little change, here a little change, here no change at all. So you choose your peak and decide what's happening. What we found is radial decrease as expected. It's a single molecule. We're talking about phase change. Why would single molecule have an abrupt transition? Just what was bothering me. What we found is, so there's hydration change of the properties. And that's kind of shows you number of hydrogen bonds between peptide and water, and number of water molecules in the shell, the hydrophobic effect. And we can then separate them into clusters and say, okay, really transition temperature for a single molecule is like this. What happens if I approximate all of the dihedral angles? It's transition from that type of major structure to that type of structure. That's what happens upon this beautiful change in color and opacity. Nothing really, right?
Nice.
Now I'm gonna skip to coarse grain and show you that if we go into coarse grain, all of the sequence dependence becomes about the same, because the biggest thing was ELPs. You can have VPG, VG, VG, PVG. There's a different behavior. Just two amino acids switched. All the coarse grain simulations will show us beautiful clusters. It's a coarse grain dynamics, state-of-the-art force field, nothing. So we did all atom simulations. This is 27 nanometers. We selected two different sequences, VPG, VG, and VGP. Then repeated by just 18 times. And now we can see some difference. Radius of duration is the same, and to distance, low difference. So entanglement inside the cluster is different. What else is different? Number of interchain hydrogen bonds. We also see some difference in number of trapped water hydrated states in them. And that's possibly can be linked to difference in experimental observation. That will. Anyways, notice that our dramatic, what we're gonna do, we all do this. I just shoot myself in the foot. I want to explain something. I say, I'm gonna simulate single molecule, and I explain to you the self-assembly. So here, single peptide infinitely dilute. That's properties, radius of duration, number of turns, fraction of, okay, all of this. Different sequences, and this is an aggregate. And this is a change. So what we see that we cannot really explain aggregation or materials formation from a single chain parameterization. It doesnn't happen. And that's an interesting part. All right, so in the other type of projects we did something cool. We're starting to predict refractive index maps from atomistic simulations. We developed a method. You can see this is work with Alon Gorodetsky and reflectins. We predicted single structure. Look at this. We have a nice CD agreement. we like to work with things that has no structure. So you notice that that's a blob, and the blob agrees, and after that, we can take this blob, in either coarse grain or atomistics, we form the clusters, then we can calculate based on volume density and all kind of different things. The aggregation here and explain the change in the refractive index as a function of aggregation, for example, and secondary strategy. All right, magnetic nanoparticles look like this. Now I'm matching all the beautiful pictures, right? Kind of close. Notice what we do. This is Vladimir Sukruk, exactly the observation of cellulose nanocrystals, different volume percent of iron oxide nanoparticles, 3.3 nanometers. This is magnetic force field. This is our system setup. Right on, 1-to-1 with experiment, just for fun, just because we can. And then we can show what happens here, right? With simulations, what we found that this aggregation clustering is critical. If it doesn't happen, it does not rotate under magnetic field, nothing wrong. Because magnetic nanoparticles has to aggregate together in order to cause the momentum change within different things. Okay, so fun and games. I actually don't do anything. Generally, all of this work is done by a couple of people. Thomas Deaton, about to graduate, Hoshin Kim, now PNL, now Lee, now Johnson & Johnson. So that's a 3 students work approximately. I can tell you, I can stay here for another 20 years and tell you about other students' work. But that's kind of one of my favorite babies because there's so many questions. And we do need community to participate in it because I cannot solve it by myself. And even like 3 or 4 of us cannot do it. We need more people. There's discoveries to happen there. There's so many questions, as I just showed you in polyelectrolytes. It's simple. And you take something else, temperature responsiveness, and it's another can of worms. It's even worse. And it's all based on biopolymers. Another thing that comes from our group, someone a long time ago, I think you here asked me, so why can't you use polymer physics for biopolymers? Like, what is it? Why is not? Why not? And I worked on that, I was assistant professor, and the first time I came, I was like, I don't know, but because it's not, right? It doesn't work. You know, look, I showed you RG, you know, like all kind of things. It's because everything in biology is directional, and in synthetic polymer is not. Very simple. DNA is directional, protein is directional. Synthetic polymers, no. Polymer physics explains synthetic polymers. You don't need to worry that hybridization versus that hybridization. So that's kind of the end of my talk. Thank you so much for your attention.
Thank you very much. It's always impressive, so many things you work on. So, time for questions for you?
I have a question about the elastin-like protein project. So I'm curious if you considered or you comment on like elastin-like proteins generally adopting like a triple helix like structure, and if you like consider that in your simulation, maybe that's like
you talk about elastin-like proteins to beta barrel. That's an Ori's model, right? It forms like some out of beta thorns. That's elastin. It doesn't form, collagen forms triple helix. Elastin, no.
Okay.
It's a liquid-liquid phase separations forms this coarservates that we try to simulate. It's like, you know, buoy type of thing. Another very interesting part about this aggregation of molecules, that if you want to compare secondary structure change, of course, the molecules inside will change differently than the molecules outside. And in experiments, you take that type of, and we take that type. And so it's like a different type of comparison that you can do, but it doesn't form triple helix. It's actually forms this buoy, messy thing that has a small rotations, violins in, violins out. I didn't show it, but that's it. So it's like the temperature increases, violins go in, temperature decreases, violin. That's it. So, and that's very prominent behavior. I think this is it, right? So PVG, so violin 1, violin 2. This is GP, of course, proline forbids you rotation, right? So it doesn't work. But you can see that's the difference as a function of rotation. So very small changes leads to dramatic things on the macro level. And that's what's the cool thing about responsive materials. Tiny, tiny alternations, huge changes in the properties of materials.
It's all Martini, right?
This was all atom, this was
All
actually,
atom. I see, I see.
Martini didn't work for, ELP did not work with coarse grain. Because we need to capture
The
that.
sensitivity, chemical details also.
This-
Actually, I was going to ask you a question about this. So I have never worked with BPP. So what is the most important thing that we miss in BPP, or what is the system that you wouldn't try BPP for?
When you have two opposite charges,
Okay, that's because.
positive and negative, everything is repulsive, everything is
Okay, I see.
negative, there's no attraction, so everything is
I
repulsive.
see, I see, I see. Yeah.
There is a new method for DPD where they included electrostatics, but then your system size immediately became coarse grain. So then it's just a thermostat that you use as a stochastic, no processes,
I see.
samples, no conformations. But I would probably stick with coarse grain and D if I use opposite
Right.
charges.
Good point, very good, okay. So any more questions? I know we are a little bit below about the 4 o'clock.
So just that you put so much work, your team put so much work in the development of the parameters for checking the DNA parameters, but it seems that we're now close to almost conditions that we can do them in cell, right? So the crowdedness level, the fraction of volume, so you think that basically, force fields are kind of getting there now.
I think so.
Okay.
I think we're also coming
That's
to the
right.
scale, right? Otherwise we don't need coarse grain as
Yeah.
much. We can go all atom.
Yeah,
Like, the reflectin, we have 64, 230 amino acid chains in the water box.
Yeah.
And we simulate it for 2 microseconds, and then we can look at the aggregation.
I see.
That's already statistics that is relevant, right? So you can actually now start comparing this experiment to a better way. So atomistically, we're approaching, I prefer atomistic simulations. We do a lot of machine learning, but only to assist us.
Okay. What is the largest system you simulated?
That's probably the one that we
The
have
one you mentioned.
64, but we work with intrinsically disorder, there's no structure, low structure,
Sure.
right? Metastable proteins, that's kind of my favorite
Right.
baby to build in the world of proteins. So they converge very slowly, generally, because they all over. You look at a simulation, they stretch, they do all kind of different things, they're looking for trouble.
Absolutely.
But that's probably the largest one, 64 times 230,
Amino acids.
yeah,
In solvent or implicit solvent?
explicit solvent.
Explicit solvent, okay.
Explicit solvent, because very often explicit solvent doesn't work in cases when you need to have hydration. So for example, coarser rates, ELP, it has to be hydrated to capture the difference. There's water stabilizing hydrogen bonds inside. So if you don't have it, there's no stabilization.
I see. I have more questions for you, but I can ask you later. Any more questions? If not, let's thank the speaker.